Thank you so much for the opportunity to participate in this session. I have no disclosures to report. Well, it is an understatement to say that we are turning the page to a new chapter of CF trial design. For those of you who grew up in the 80s, not to date myself, but you may remember those Choose Your Own Adventure books. I think that's a perfect analogy of where we are at today, as we need to explore who are the new target populations for development, what trials are the CF community interested in participating, and how are we going to design efficient trials across the CF population now divided by modulator hues. It's really the combination of the who, what, and how that is going to define our future therapeutic pipeline. So as daunting as that may be, we need to remember that we are the star of the story as we embark on this next chapter. So let's start with the who. So of course, we are developing therapies for all individuals with CF. However, we do need to recognize that drug development efforts are streamlined by focusing on populations for which we are most likely to detect a meaningful clinical benefit, hence the definition of a target population for development. So on the left-hand side, I have promising classes of therapies that are in our development pipeline. And along the top are potential target populations defined by modulator eligibility and access. Now along the left-hand side are potential target populations without modulators, and then those with modulators, maybe with or without more symptoms now that they are on modulators. So we can start to naturally define target populations as we go forward through development. For genetic-based therapies, of course, the most critical development is for those, that 10% of the population without access or that don't have these modulators available to them. But we need to start asking some tough questions here in terms of whether or not that remainder of the population could help in these development efforts. Perhaps providing early safety data, or in the case of mutation-agnostic gene therapies, when do we start to bring that development into the modulator population as they may also benefit from these therapies? For new CFTR modulators, of course, it would be easiest or ethically easiest to develop these therapies among those who are eligible but don't have access. But again, how can we include those who are modulators in this development? And I think we need to recognize that for symptomatic therapies, we would expect, greater treatment response among those who don't have modulators. But, those on modulators, there are likely some who can really benefit from these therapies still. So as we step back, we've got a checkerboard here. If you're confused on the greens and yellows, that's okay. That's the whole point of this little exercise that we are not now faced with. But there is a little bit of clarity in those green dots. We know we need to focus efforts, and there is a clear development pathway for those that don't have access to modulators. But we have to be a little bit careful here. The numbers are so small. We just take with the US, the number eligible, and if we filter that down to the number of adults who would qualify for, those early phase studies, and then factor in our historic trial participation rates, we're really talking just the low hundreds of accessible patients for our trials. And so if we think about a robust pipeline of both symptomatic and gene therapy programs that we are trying to move along, we need to be very careful that we're not, expending this population, this precious population. And we try to move all of these therapies forward. So not only do we need to make sure we are doing development, for all of our CF patients who can benefit from these therapies, but in particular, including the modulator population in drug development efforts will be absolutely necessary to move multiple therapies through the pipeline for the non-modulator population. So what we're trying to do here is take something that looks like this and turn it into something that looks a little bit more like this in terms of our drug development efforts. Now, this is all well and good, but it relies on the fact that we have an engaged community for doing these trials. And so this is really where we need to step back and pause before we get into the how and ask, what trials are the CF community interested in participating? And in particular, those who are established on highly effective modulators. Before we look at this a little bit more in depth, I think we need to challenge some of our natural associations here that the need for therapies may be directly correlated to clinical trial interest. We may have, a modulator population that may not have an acute need for some of these therapies, but a high clinical trial interest. We're actually starting to gather some of this data just within the last few months that I'll start to review with you. So we have an ongoing study called CheckFC, which is actually enrolling patients when they've been on highly effective modulators or on CFTR modulators for at least 3 months. And we surveyed this population, and again, this data is just collected over this last year, up to almost 1,000 patients, on their willingness to participate in future modulator trials, where these modulators are hypothesized to be as effective as an ETI, for instance. Well, you can see here that there is relatively high engagement. I think this, continues to surprise us. Of course, higher participation for active comparator studies, but still, reasonable interest in participating in placebo-controlled trials as long as they're short-term. Now, it's worth noting that we asked for longer duration trials, but we likely would not ever consider those for the concerns of safety. And we should point out that, there is still, the impact of highly effective modulator withdrawal with recent reports coming out really question our ability and timing of how long we could, in fact, do any sort of placebo-controlled trial. It may just be a week and not a month to be able to do those trials safely. But all in all, there does seem to be engagement from the community. This similar trend was seen as we asked the cohort of those who are on chronic anti-pseudomonal antibiotics their willingness to participate in future inhaled antibiotic trials. And what we'll see here, these are designs that would include a placebo control. Again, this idea of support for shorter term studies. And of course, this tracks well with, those who would be on intermittent therapy. So there's clear engagement there. We do note in this setting that there are some caveats of how we would enroll these patients who are on ETIs. We're starting to see, issues such as sputum production coming into play for eligibility for these trials. And a parallel survey, however, from the CF Community Voice Group was able to demonstrate that approximately 70% of about 56 patients surveyed produced a dime size or greater sputum sample in the prior 2 weeks on ETI. So I think this is important because it's showing that we may need some flexibility here, and especially when it comes to home collection and windows around our clinic visits instead of this on-demand sputum as we're thinking about these types of trial designs. We're seeing similar interest in genetic therapy trials. Among approximately 370 surveyed through CF Community Voice taking CFTR modulators, the majority were interested in participating in these trials, even though they were on modulators. So I think, lining up, this data is all lining up with our strategy, but our community is really challenging us here to think creatively, be inclusive, and offer opportunities in our drug development strategy, regardless of modulator status. So then that gets to the how. How will we design efficient trials across the population now divided by modulator use? So let me walk through a couple of challenges here when it comes to study design. So the first challenge I want to talk about is when we're expecting differential treatment effects across the population. So let's talk about an example. Say we have a new pulmonary therapy candidate. And we might expect that in the population who are on highly effective modulators, we may not expect more than, say, a 3% improvement in FEV on top of modulators. However, in those who are not on highly effective modulators, we would expect a larger improvement, maybe a 5% or more improvement in FEV in the absence of highly effective modulators. So what are our options for trial design? Well, we could do a randomized trial exclusively in those without modulators. The average treatment effect maybe is 5%, and this would take about 62 patients. So not huge, but again, the overall population is relatively small, not even factoring in historic trial participation rates. The other option is to do a randomized trial in both populations. Say we have about a 4-to-1 ratio of those who are on modulators to off modulators. This approach would attenuate our overall expected treatment effect to be about 3.5%. We'd increase our sample size because of this to about 114, so not huge, relative to the massive increase in our patient population that we could now recruit from. So these are the types of things and exercises that we're now going through in terms of our drug development strategy. The question here is, with that smaller treatment effect size, is 3.5% meaningful without supportive secondary data? Typically, the smaller that FEV effect is, the more we've had to rely on some of these secondary endpoints like exacerbations to really solidify the efficacy of a therapy. We may no longer be in that position to rely on some of that secondary data. If we look at exacerbations, we can just see the dramatic decrease with ETI, annualized rates of 0.37, that may be even lower with reports of the longer-term follow-up data of these patients down to, say, 0.3. So exacerbations may no longer be a viable outcome for CF. If we were to power a trial at this endpoint, we would need more than 700 patients followed for a year to see a 40% reduction associated with the given therapy. So really, one of our pathways here is to really stand behind FEV, and these more moderate effects are important, particularly when we see some differentiation in the population and knowing that, we need that modulator population to help establish those effects in the non-modulator population. But we do also need to think about moving beyond FEV and exacerbations. And so what is the work ahead? In simple terms, we have a couple options here. We could, look towards elevating some of our current endpoints that we have in our toolkit, LCI, imaging, maybe biomarkers, to be more in that position of a surrogate endpoint for clinical efficacy. Now, we need to define what they're a surrogate for. Is LCI a surrogate for exacerbations? Is it a surrogate for FEV? There's a lot of work that's going on to really link these outcomes to key clinical efficacy measures. The work ahead is we do need more bridging studies to really establish that the intervention's effect on the surrogate accurately predicts the effect on the clinical outcome. So we need more studies that are including all of these measurements, our trusted endpoints, with these, what would have been secondary endpoints in our prior trials. The second option is to create new endpoints, entirely new endpoints, maybe a fit for purpose endpoint that's specific to a particular therapy. A new PRO, maybe a new exacerbation endpoint. That is actually a subject for an entirely different talk. This create path does require a whole validation process. It could take years. And so that is something that is in our wheelkit, but there is an extensive validation process as well with this path. Whichever option we choose, it's likely that we're not going to be able to rely on just one study to provide the evidence for these outcome measures. We're going to need to rely on multiple studies. So I want to recognize the group of our colleagues in the UK on the Core Outcome Set Task Force for CF. Their efforts are really to have stakeholder engagement to define core outcomes across indications for our trials and ensure the collection and the publication of these outcome measures consistently. this is key to our strategy to ensure that we have a foundation that we can do meta-analyses across our studies and really elevate and create and validate these endpoint measures moving forward as we redefine the disease for this new study. Our second challenge here is when the development of a new therapy requires withdrawal of standard of care, most often seen for anti-infectives or modulators. We talked about the potential here. We may have a window to do a short placebo-controlled trial. For modulators, it may only be a week or so. This is, a strategy, but it may fall short to acquiring some of those key safety and efficacy parameters that regulators like to see. So we are going to rely on a piecework of studies to really get to that package, including open label active comparator trials. Regulators like to see blinded studies, but likely we're going to have to rely on open label studies in these scenarios for practicality purposes. And also the role of historically controlled comparator studies using real world data to help boost this as well, moving what typically was used as post-marketing studies more into that regulatory package to meet all of our criteria. Our third challenge that I want to talk about is when the small non-modulator population is the target population for multiple development programs. And we want to move, multiple shots on goal for this population. So what are the levers here to increase those shots on goal in this population? Well, the first lever we have is really to just increase our trial participation rates. 40% of individuals with rare and non-sensitive mutations are people of color. So we have a disproportionate number of these patients in this trial, in this population. We need to understand and reduce barriers to enrolling historically underrepresented individuals with CF in our studies. This is critical to our path. In a recent survey of our research coordinators in the TDN, we actually, evaluated what these barriers are and identified, language and cultural barriers are the most common barrier we are encountering. But there are also some practical barriers such as transportation, time, having weekend visits. So we systematically need to go through these barriers and really knock them down as we move forward with our strategy. The second lever we have is really to promote innovative designs that decrease sample size burden across this population. One idea here is the use of synthetic controls to decrease or replace concurrent placebo groups. Each study that we design, we're not going to be able to do those traditional placebo-controlled trials. And so how can we rely on some novel ways to establish a control group without utilizing our current patient population? So the idea here is that we could use either real-world data or historical trial data to match, with our concurrent active patients. Now, this strategy has been used in oncology mostly, but it's also gaining traction across other rare disease settings. And in fact, a recent article really was just promoting this strategy and guidelines in regulatory agencies as part of the 21st Century Cures Act. We are actually developing methodology, evaluating our outcome measures of how we can do this within CF and establishing proof of concept for this. I refer you to poster number 47. And the TDN is actually exploring accessible and regulatory compliant data repositories of completed clinical trials to offer as a resource to sponsors for these innovative trials. Now, there is a little bit of a catch here, because this does require data sharing across both academia from our investigator-initiated studies and industry. Sort of this give to get kind of idea in order to get there. So I'm quite confident that we are going to get here to really involve and include our patient population. And I just want to say that our adventure awaits, and we are not alone in our effort to, develop this new chapter. So thank you very much.
Hello, my name is Jennifer Groski. I'm from the University of North Carolina at Chapel Hill, and I'm going to be speaking today about functional imaging approaches to ventilation analysis. I have the following relationships to disclose related to this presentation. I've received research grant funding from the Cystic Fibrosis Foundation and the NIH, and I've also received consulting fees from the CFF and Polarin. The question facing all of us as clinical researchers is, in the era of highly effective modulator therapy, how do you detect and monitor mild CF lung disease? So certainly we know that we can measure lung function routinely, and I think all of us are fairly familiar and comfortable with the use of spirometry. One of our later speakers is going to talk in depth about multiple breath nitrogen washout, which is gaining traction as both a clinical and research measure. But these don't offer any information regarding the structure, the anatomic structure of the lung. Now, we can look at the anatomic structure of the lung using chest X-ray or chest CT, but chest X-ray, although widely available, really is fairly low sensitivity for detection of CF lung disease. And chest CT, while also broadly available and in routine use in Europe for both clinical and research reasons, does carry with it some risk related to ionizing radiation exposure. There are some newer methodologies that are currently being explored, such as capture functional small airways disease via biometric response mapping, as well as other methods to decrease radiation exposure. Earlier this year, the Cystic Fibrosis Foundation hosted a virtual workshop called the Imaging Outcomes Needs Summit, where we reviewed existing and upcoming image modalities. And a lot of discussion actually focused around MRI techniques. Key discussion points included how to standardize across sites, the creation of data repositories, the possibility of artificial intelligence to remove reader bias, and leverage of free data from surrounding structures. When you image the lungs, you don't just see the lungs, but you also see bones and muscle and vasculature. So you may be thinking to yourself, MRI for the lungs? Really? Well, we all learned in medical school that conventional MRI uses radio frequency pulses to manipulate nuclear spins of protons and capture the resultant resonance signal. This has traditionally not been a good approach to take in the lungs because of fairly low proton density. However, you can see by this histogram taken from PubMed that the research that has gone into improving MRI over the last several years has really ballooned. And I've listed just a few of the relevant publications. By adjusting the sequences and other parameters used to acquire MR images, we've been able to show that MR can detect structural abnormalities in preschool children, that it correlates with high-resolution CT findings, and that it's responsive to changes in disease state, such as showing changes with treatment of an exacerbation. And even the Fleischner Society, which is my go-to every time I encounter a pulmonary nodule, has recently published a position paper. This came from Chest in 2021. And in it, they reported that they felt there was good evidence for clinical use of pulmonary MRI in the CF population due to its ability to detect mucus distribution, bronchiectasis, inflammatory air wall thickening, and among other changes. Here's two images taken from a recent radiology paper. Ultra-short echo time images have really vastly improved how we're able to detect CF-related changes in the lungs, such as bronchiectasis and airway wall thickening. The top image here is a UTE image. Down here is a T2 weighted image. And there are also zero echo time images that are currently under study that allow us to detect typical changes that you might expect to see in the CF lung. So MRI actually has a lot of opportunities available to us. It is widely available in academic medical centers. We have seen that it is able to detect subtle anatomic and physiologic changes. And again, we can build AI algorithms to eliminate reader variability. There are, of course, limitations. There can be long scan times, it's relatively expensive, and there's differences across the platforms by different vendors. But sometimes when you look at an MRI, to the untrained eye, it can be difficult to distinguish between MRI and CT scans. This recent publication from CHEST shows MRI images across the top in panels A, B, and C, comparing them to CT images in C, D, and E. And here you can see that both methodologies are able to detect saccular bronchiectasis, mucus plugging, and even consolidations. Furthermore, there's been extensive effort to be able to score MRI scans in order to achieve the ability to compare across different sites. This is one such scoring system that's probably the most widely used one. And here you can see that the reader will evaluate for bronchiectasis or wall thickening, mucus plugging, abscesses or sacculations, consolidations, special findings, and perfusion defects in the right and the left lung by lobe. And the larger the number, the score that's given, the worse the disease. And so some have even gone to the point where they've actually used paired MRI as a clinical trial endpoint. Another publication just this year looked at adolescents who were treated for anywhere from 2 to 4 months with lumacaftor or ivacaftor. And they did baseline MRI scans on them and then compared them after treatment. And so what we can see in some of the imaging here is improvement in the bronchiectasis and the mucus plugging, as well as consolidations that we had seen on the baseline imaging. As well as there's improvement in perfusion defects, suggesting that there's better ventilation to those areas as well. And this is important because in the same study, FEV1 was not a clinically significant finding in terms of comparison from baseline to treatment with lumacaftor or ivacaftor. Here we can see the individual scores down here showing the morphology improvement, perfusion improvement, and global score improvement. Now, I want to just briefly touch on oxygen-enhanced MRI. This is a technique that's been around for over 20 years, and it relies on the fact that there's a shortening of the T1 relaxation time of 100% oxygen relative to 21% oxygen that's found in room air. And it uses subtraction analysis in order to be able to grade areas of ventilation defect. And these are outlined here in the green and categorized as VDP, or ventilation defect percentage. And so here are 2 examples of relatively severe CF patients with substantial ventilation defects. But one of the most interesting and promising new technologies is actually hyperpolarized gas MRI. In this scenario, we use an inhaled contrast gas in order to highlight the lung spaces and be able to tell us information about both ventilation and structural lung disease. The benefits to using hyperpolarized gas MRI is that using an isotopically enriched gas can improve the signal to noise ratio and allow you to detect disease better. And in particular, most of the world is using hyperpolarized xenon MRI currently. And in particular, hyperpolarized xenon has particular unique chemical shifts that actually allow you to quantify gas exchange by having different peaks in the gaseous phase, the interstitium, and the red blood cells. And you can truly get then at the diffusion of the gas into the cells. There are limitations, of course, that exist with hyperpolarized gas MRI. And primarily among these, it's the lack of standardization across the platforms, the centers, the protocols that have been performed. But it also relates to the short half-life of the hyperpolarized gas in that the longer it is exposed to oxygen, the less polarization there is, and thus the less visualization of ventilation defects. Here's an example of a CF patient before and after being treated for 1 month with ivacaftor that was published in JCF in 2017. And you can see the areas of ventilation defect that have nicely recovered after treatment with modulator therapy. So in order to address some of the limitations with hyperpolarized gas MRI, there several centers with expertise in xenon MRI have come together to form a clinical trials consortium. And here they are across the country in the US. And they recently published a position paper in MRM. And in this paper, they discussed what the most appropriate protocols and techniques would be in order to be able to standardize using hyperpolarized gas MRI to image ventilation using multi-site trials. So this is really a step in the right direction in order to be able to perform multi-center clinical trials. And in fact, these centers are currently performing a study called the HighPoint study, Hyperpolarized Imaging for New Treatments, that can be found in clinicaltrials.gov. So one of their studies thus far was a 2-center comparison. The group at Cincinnati and Toronto individually imaged with hyperpolarized gas MRI, imaged CF and healthy patients. And then they took all their data and they had 2 independent analysts review data from both centers. And they compared the outcomes of those reviews. And so you can see here the anatomic image overlaid with the hyperpolarized gas image from the 2 different centers. And the pink color here depicts where the ventilation defects were located between analyst 1 and analyst 2. And there was actually a really tight agreement between the 2 analysts overall in what constituted a ventilation defect. They were also able to distinguish a significant difference in VDP between healthy controls and CF, even when the data sets were combined. And there was good inter-reader correlation as well. So all this has actually pushed the field of hyperpolarized gas MRI forward and towards FDA approval. So there has been a new drug application that's submitted to the FDA for use of 129 xenon for pulmonary imaging. And by the time you watch this video, hopefully there will be a decision regarding this, as the target decision date is in early October. Now, my particular research team looks at a different gas called perfluoropropane, and we are exploring perfluorinated gas MRI for use in CF. The major difference between these gases and hyperpolarized gases is that these are thermally polarized gases and are commercially available in large quantities. And so that allows us to provide a multi-breath procedure and dynamic evaluation of ventilation as a subject breathes the gas over time. So here in the center, we have a healthy control patient who is breathing the gas. And as the subject breathes the gas in, you can track each individual voxel. And if you were to plot that particular voxel over time, you would see as gas washes in during our breathing in portion and then washes out during our breathing out portion of the multi-breath protocol, you can track that voxel intensity over time. And that allows us to estimate what the slope of this gas wash in and wash out phase would be, and compare it between healthy controls and patients with CF. And here you can see at the end of washout that there's some evidence of trapping of gas here, that the gas doesn't fully leave the lung. This is the setup that we use. We are studying them in a 3T MRI with a 19F tuned chest coil. And here you see a bag of gas that they're breathing in through a one-way valve and then breathing out. And so we start off with a proton scan, and we've since moved to UTE scanning as well. And breath by breath, we can analyze. And I've only included a couple of the images here, but you can see as the first breath of PFP goes in, what it looks like at the final breath with some obvious ventilation defects. The first breath after we start to wash out the gas with room air, and then a final washout. We've come to some interesting conclusions regarding using this method and how it compares to other current available methods. First of all, we compared healthy controls against mild CF patients with normal lung function and moderately affected CF patients with FEV1s around 60%. And in this study, we showed a difference between all three subgroups, and particularly between healthy volunteers and mild CF patients, in the fraction of lung volume that had slow gas washout time. Importantly, VDP did not distinguish between these groups, and neither did FEV1. We subsequently looked at 19F-MRI directly compared to xenon MRI. And here you can see two images side by side. This is 19F-MRI, and here's xenon MRI. And clearly, there's improved signal to noise in the xenon scan, which was expected. However, what we found was that the ventilation defects acquired with dynamic versus static imaging are not identical. And when we looked at the shape of that gas wash-in and wash-out curves, we believe that we can explain that by the difference in slow versus fast ventilation, such that there are some areas that end up showing the presence of PFP gas, but don't have xenon gas. And so rather than being a true ventilation defect, these are likely areas with slowly filling regions. We've recently completed a study, a small pilot study in 8 patients, studying them before and after initiation of elexacaftor, tezcaftor. We were stymied by the pandemic, of course, unfortunately, and so we didn't enroll as many as we hoped to. But we did look at these 8 patients before they started the drug and after 1 month of treatment. And we're gratified to see our expected changes between baseline and post-treatment changes in FEV1 and also improvements in LCI. And when we look at the abnormally ventilating components of lungs, including those that constitute ventilation defects and those that constitute slowly emptying regions, we also saw a significant difference pre and post-treatment. Importantly, we experimented with using ultra-short echo time MRI as well and had 2 independent readers review these images. They were blinded to the stage of disease when they were reviewing these, and they found a significant improvement in the scores that consisted of bronchiectasis or airway wall thickening, as well as mucus plugging after 1 month of treatment. So obviously there's a lot of different methodologies that we can use, and it's not clear which of these is going to end up making its way into clinical trials or if it's going to be some combination of both. But there are many reviews that are out there now comparing and contrasting the pros and cons of each of these different types of images. So I hope I've convinced you that for future considerations for clinical trials, it's important to think beyond FEV1 and to think about all the different techniques and what we're actually interested in learning about the ventilation of the lung. Particularly important key areas to explore include standardization across lung volumes as children grow into adults, how we standardize multi-center protocols, and how we automate image analysis. With that being said, thank you for your attention. Certainly, this is a product of team science, and I'd like to thank all the collaborators that I've worked with over the years. I'd be happy to answer any questions during our open Q&A.
First of all, I'd like to thank the organisers for the invitation to talk today. My name is Clifford. I'm a research respiratory physiologist and PhD candidate from Imperial College London and the Royal Crompton Hospital. I also work on behalf of the ECFS CTN LCI core facility under Professor Jane Davis and Claire Saunders. These are my disclosures. And the talk I'm going to give today, I'm going to briefly introduce multiple breath washout and what it is, why is it needed and how does it compare to other biomarkers, the considerations and limitations when performing the test, and the minimal clinical important difference and has it been established, how has it been used as an outcome measure in clinical trials, the most recent developments, and is it ready for prime time? So multiple breath washout is a measure of ventilation efficiency. It's often quoted in the literature as a measure of ventilation inhomogeneity, but it also measures physiological dead space and gas mixing efficiency. Conventionally, it only measures the communicating lung. So if anything is blocked off via a mucus plug or is collapsed due to lung disease, then that won't be measured. It's an inert gas washout, so you have a couple of options. You can either wash in a gas into the lung, such as sulfur hexafluoride, and wash it out with room air, or you can use the resident nitrogen within the lung and wash that out with 100% oxygen. It's a tidal breathing test, and lung clearance index, LCI, is the main outcome that we use. So this is an example of a nitrogen washout. In the top red line, you have volume, and behind that in black, you have flow. In the burgundy line, we have our nitrogen trace. We establish relaxed tidal breathing, and then we turn on 100% oxygen. And then during inspiration, that goes down to 0%. And then during expiration, you get each one of these little peaks. And breath by breath, we go all the way down to 1/40th or 2.5% of the starting concentration. Each one of those little peaks adds up to the cumulative expired volume of nitrogen or tracer gas, divided by the functional residual capacity, gives us our LCI, and this is a nice healthy subject, so they have an LCI of 7.78. This is a adult cystic fibrosis patient, and if you notice compared to the other patient, the time axis down the bottom, it only took 180 seconds on our last subject, but this one takes 270 seconds. They had a much higher cumulative expired volume and a smaller FRC, so their LCI is 14.04, quite a severe abnormal LCI. So why is it needed? So could we just rely on spirometry alone as the gold standard? Unfortunately not. If you want to measure the health of the airway tree, unfortunately spirometry just measures the health of the airway trunk. This is because FEV1 is dominated by the large airways, as it's a measure of volume, and as little as 10% of the signal from FEV1 comes from the peripheral airways. It's age and effort dependent, and it's also quite a poor predictor of mortality or lung transplant, just as good as flipping the coin, which has led to the so-called silent years we see in cystic fibrosis over the last few decades. And then alternative lung function tests such as body plethysmography and gas transfer, unfortunately, do not provide any short or long-term prognostic value in cystic fibrosis. So how does LCI, our main outcome, compare to other biomarkers? So in comparison to FEV1, we've seen large, significant changes in LCI, whereas there's been little or no change at all in FEV1. And we've also seen those treatment effects happen when FEV1 is within the normal range. If we just relied on FEV1 alone to determine lung health, around 53% of the patients we test will be wrongly classified as normal. In comparison to CT, there's often a good correlation between LCI and the total lung scores, and specific scores like bronchiectasis and mucus plugging. However, there's a bit of a weaker, but still a moderate correlation with air trapping, and that's to do with the non-communicating lung portions, which we talked about in the introductory slides, not being measured. There's no correlation with the parenchymal score, and they're considered complementary markers of disease severity, but one cannot replace the other. An alternative to CT is MRI, which can be used just as an image like CT, has again good correlation with global scores and specific morphology scores. But there's also another component to MRI. You can have a functional MRI, which you can either use a hyperpolarized gas, such as helium or xenon, or a project which is just being funded by the Cystic Fibrosis Foundation and is due to get underway any day now at the Royal Brompton Hospital, is looking at oxygen-enhanced functional MRI. And there's an example here on the bottom right-hand side, and we see in blue the ventilation defect percentage, which is one of the main outcomes from oxygen-enhanced MRI. So what we know. So the minimal clinical important difference is yet to be defined for all age groups. And the clinical change, or the clinical significant change, was always thought to be around 1 unit. But recently, a randomized controlled trial showed no benefit to patient outcomes when they used the 1 unit threshold. The natural variability is not yet defined. The best attempt has been done at preschool, and then further attempts have been done at school age. The reason why the 1 unit change is not particularly appropriate is that as the disease severity increases, so as an LCI increases, so does the standard deviation between stable visits. Therefore, a fixed percentage change is recommended instead. And in preschool, there was a real good study from 3 CF centres across North America, where they recruited 71 healthy controls and 77 cystic fibrosis patients. They had over 500 visits over a 2-year period, and they found that that minimal clinical important difference was around 15%. So that's always derived from the healthy control to see the actual change of the test. If you just did it in the cystic fibrosis population, it would be much higher at 25%. The school age studies from Gothenburg and Copenhagen, they did not use healthy controls, so we don't have that minimal clinical important difference. But they did find an increase of 17% compared to the previous LCI value, may indicate lung disease progression, and that was from Gothenburg. And in Copenhagen, they found something similar of around 25%. 25% change was physiologically relevant, but again, we need those healthy controls for a true picture there. So multiple breath washout in clinical practice, this has really boomed over the last 5 years. So now there are more than 20 sites across Europe where it's performed at either annual assessment every 6 months or in some cases every clinic. The predominant device is the Xlizer D, which is a nitrogen washout. But the consensus from the operators and clinicians using multiple breath washout is that before it can be used optimally and in widespread clinical practice, that minimal clinical important difference is essential. So how is it being used in clinical trials? So we found in CF that it can be abnormal in infants, preschoolers, school age, and adults. It's successfully encroached on the so-called silent years brought on by FEV1. And we know that abnormal preschool LCI predicts FEV1 decline later in school age. So the early studies of hypertonic in DNAs, so they found that 4 weeks of twice daily inhalation of hypertonic saline compared to the active placebo of isotonic saline improved LCI, so a decrease of 1.16 units, whereas there was no change observed in spirometry and quality of life. They also saw a significant treatment effect in preschoolers after 48 weeks. Again, in the 16 to 18-year-olds age group, they found an improved LCI versus the placebo of 0.90. And then probably one of the most important studies, which kind of changed the landscape of how multiple breath washout studies are performed. This was done in subjects who were 8 years and older with a preserved FEV1 of more than 90%. They saw a very large decrease of 2.2 units in LCI. But this was a multi-center study. And importantly, they had a mix of naive and experienced MBW sites, which showed that the naive sites could be trained and they could get good quality data to be used in clinical trials. This led to a call of standardization with protocols and different device and to use the same device. 3 central overreading centers were set up. So Sick Kids out of Toronto in North America, led by Felix Ratchin and his group. On behalf of the ECFS CTN, Professor Jane Davis and Claire Saunders set up the Lung Clearance Index, where I currently work. And Paul Robinson set up the Australian Central Overreading Centre at the Children's Hospital in Sydney in Westmead in Sydney rather. And we've worked on a number of trials now, but our first big one was the study of lumacaftor and ivacaftor in school-age subjects 6 to 11, where we saw a large decrease of 1.1 units. A preschool study of ivacaftor with just 9 subjects saw a very large decrease of 2.7 units, and that was over 3 months, but then sustained over a 6-month period. An Israeli study saw a decrease of 0.68 units compared to the placebo, and that was coordinated out of the Royal Brompton Hospital. And then recently we published our tezacaftor ivacaftor study. Again, in 6 to 11-year-olds, we saw a decrease of 0.51. And finally, our most recent paper the paper of the triple combination, elexacaftor, tezacaftor, and ivacaftor, saw again a large decrease of 1.71 units in the 6 to 11 age group. But there have been a number of studies where LCI has been used as an outcome, but there has not been a significant decrease. However, there's often been no change in the other outcome measures that were used. The limitations and considerations when performing or when using the tests. So the main reason why the central overreading centres were set up is because quality control is absolutely essential. It's a sensitive test, so we need sensitive operators to be performing the test. And tests with leaks are not valid. It's a tidal breathing test, so ensuring that is absolutely vital, as this can have a large impact on the functional residual capacity and therefore will affect the accuracy of the LCI values. And then even more so when multiple breath washout is used in clinical practice, operators should be trained, experienced, and skilled, especially if those tests are going to be used for clinical decision making. LCI for tracking severe disease, unfortunately, is rather time consuming, 1 to 2 hours. And due to the nature of the test, as we know that it only assesses the communicating lung, we often see a paradoxical effect in severe disease, where at the baseline measurement, trapped gas is not measured, but due to the positive intervention, those areas of trapped gas have now been opened. So then in the post tests, they get measured. And unfortunately, you see an increase in LCI, so a worsening of LCI, despite an improvement in other outcomes such as FEV1. Also see the same response sometimes in intravenous antibiotics. And this also accounts for a large variability between stable visits, which we saw, and why the main reason we need to use a percent difference rather than the 1 unit standard clinical change. And we also don't know if there's any clinical difference, for example, of an LCI versus 16 or 20 or something along those lines. We also know there's potentially large differences between gases. And we know that they are not interchangeable, and even different devices are not interchangeable. Nitrogen values are often much higher than corresponding washing gases. And if you can see on the right-hand side, the black dots are nitrogen and the open dots are SF6. This was a study of simultaneous washout conducted by Dr. Katie Bayfield at the Royal Brompton Hospital. And as you can see, it takes nitrogen to get down to that target concentration for an extra 20 breaths or so for this subject compared to SF6. And this was predominantly thought to be because of nitrogen back diffusion from the tissues into the lungs when using 100% oxygen. However, a recent development has shown that this was not really the case, and that there has been a cross-sensor error in the nitrogen measurement. So basically, this led to an absolute overreading of nitrogen of around 1%, which basically caused an extended tail to nitrogen washout, which is again what we pull cystic fibrosis in healthy controls apart from. A new software released by the manufacturers and the investigators of this research has recently been released, and we think that SF6 values and nitrogen values should be a lot closer now. A number of normative values have been released, as typically you can see the SF6 values are a lot lower than the nitrogen values. But the great work that the Global Lung Initiative did in spirometry, body plethysmography, and gas transfer to create normative values, they are also going to do this for LCI values and other MBW parameters. And that work is already undergoing. There's also recently been shown to be a predictor of mortality. So this was a time to event analysis of either death or lung transplant. And in their crude model, they found that an increase of 1 Z-score led to a relative risk increase of either death or lung transplant of around 4%. But their final model, where they took in a number of confounders, they found the risk of death or lung transplant increased by an average of 30% per 1 standard deviation increase. And then recently from our lab, we've released this extension to multiple breath washout. And this was an attempt to fix that limitation of only measuring the communicating lung from multiple rest washout. So adding a slow vital capacity after the conventional end of test gives us a functional measure of trapped gas or under or unventilated lung units. It was feasible from children from the age of 5. And importantly, it could not be predicted by the baseline LCI 2.5. And an important finding, we found that both LCI and LCI SHX, our new parameter, correlated significantly with total lung CT scores and hyperinflation score, but only LCI SHX correlated with mucus plug. This is what it looks like. So normally our target concentration would be reached at the start of that red shaded zone, and then we would stop the test around the end of that red shaded zone. But now we do a slow vital capacity, and our signal is the change in nitrogen concentration. So in this case, an increase in nitrogen concentration. And we see 2 patients here, both CF females of 7 years of age. They both have normal FEV1s of 90% and 101% respectively. They have very similar LCIs of 9.3 and 9.5. This top subject, however, when they did their slow vital capacity, the nitrogen only increased by a very, very small amount, and their extent of under or unventilated lung units was only 0.3 units. So their LCI SHX only increased by 0.3 compared to their LCI, and that is measured on the same units. However, the person at the bottom, we see a very large increase in nitrogen concentration with the slow vital capacity. And then for 5 breaths after that slow vital capacity, the nitrogen concentration remains above that green dashed line, which is basically our signal. Their corresponding CTs, the top person had 0% hyperinflation, which is what we could basically predict from their UVRU score. But the person at the bottom, they had a very high hyperinflation score of around 22%. And that's also what we saw in our corresponding short extension. And then if we look on their corresponding CTs and we just fiddle with the CT images just slightly, we can see this large patch of hyperinflation on the bottom CT there. So the question is it ready for prime time / widespread clinical use? As you would have heard from my language throughout this talk, not quite. So we have a few unanswered questions. We need to figure out who and when it's used for, how often should it track pulmonary exacerbations and change in treatment. We really need to find out that minimal clinical important difference for age groups. So the preschoolers are ready to go, but we need it from school age upwards. A couple of recommendations that we have. The preschool age group could be used now, but it could be also used all the way into adults with a preserved FEV1. But if they don't have a preserved FEV1 and they have severe lung disease, then alternative tests such as functional MRI or CPET should be used instead. And because quality control is so vital for getting accurate results from multiple breath washout, and an accreditation should be available for clinical MBW operators, especially before this is used in widespread clinical practice. I would like to thank you for your attention today. And if you have any questions or would like to reach out, these are my contact details. Again, thank you very much for listening.
Organizers for the opportunity to present on biomarkers of CFTR activity. My name is George Solomon. I'm an associate professor of medicine at the University of Alabama at Birmingham and a scientist in our CF Research Center and the Adult CF Center director. The following relationships are related to this presentation, including grant funding, non-pharma grant funding, advisory board, and consultancy. This talk will discuss the following outline. We'll review the latest trends in biomarker development as it relates to potential CFTR-based therapeutics and provide a bit of an overview and summary of current and recent work. I'll explain the recent experience of biomarkers in CFTR modulator development as a background for what is going on with current path to cure and gene-based therapies. And we'll review the implementation of emerging data from early gene-based therapies and next steps for developing CFTR biomarkers for this type of investigation. So there are several characteristics of a, quote, perfect biomarker, which include reliability, several different types of high tier validity, and responsiveness, or how likely the perturbation in the biomarker responds to a therapy. Of course, there are no perfect biomarkers, but there is a large menu, which I'll review next, of both in vitro, which include collection from sites including nasal, bronchial, intestinal, and blood or skin for iPSC or induced pluripotent stem cell derivation. And then there are multiple types of analyses that can measure out or read out CFTR function either directly or indirectly. Most commonly, those include organoid swelling by forcible-induced swelling and or CFTR, other channel-dependent short circuit measurements, or ISC. In vivo measures come in several flavors as well, which include potential difference measures like nasal potential difference, or less commonly used lower airway potential difference. Of course, there's the very commonly used sweat chloride test, and there are downstream effects which are in development, including mucus clearance in the bulk level, or epithelial properties of cilia beat frequency, or other properties on the epithelial surface. So do we have an ideal biomarker, especially for the next set of trials, including genetic-based therapies? Of course, the tipping point may come in what is the most interest by sponsors, and right now appears to be related to in vivo for clinical trial development, especially proof of concept studies, because of target tissue delivery, the proximate nature of the drug administration during the trial with the measurements, and the biological plausibility and evidence for proof of concept. However, there are many important and desirable facets of in vitro biomarkers, which includes the relative standardization, the level of repeatability, and there's a significant amount of prior experience with these. There is significant interest in advancing in vivo biomarkers for genetic-based therapies, especially lung-derived delivered therapies, which would rely on probably and most likely lower airway potential difference or other measures like this. While sweat chloride has definitely been a workhorse, it does require therapy to be systemically administered to be an effective biomarker for this type of study, though. This figure by Nicole Meyer-Hamblett really encapsulates many of the properties and an association of various levels of biomarkers. As you can see, on a global level, biomarkers can be used as markers of biological effectiveness or efficacy, and really may show a change in response to therapy as kind of the lowest tier bar for the use of a biomarker for CFTR function. As we go in, biomarkers can be developed as clinical correlates, which many of the in vitro and some of the in vivo are now reaching the level of clinical correlation. And then finally, with continued development, may serve as surrogate endpoints for other clinical endpoints like FEV1. We've not reached that level of any of the biomarkers currently. As stated earlier, sweat chloride is the most advanced. It's become really a workhorse for ongoing modulator development. As you can see, there's variability in the individual correlations of sweat chloride to, for instance, key outcome clinical measures like percent predicted FEV1. However, and most recently shown in the bottom left, the PROMISE study has shown the first evidence of a direct correlation at an individual level of change in sweat chloride to change in percent predicted FEV1. Regardless if this correlation holds up in larger studies, which has not been shown across the spectrum, we do know that the extent of sweat chloride reduction does correlate to on a group-wise level in many trials, as shown on the right-hand figure, to the level of improvement of key clinical outcomes, including percent predicted FEV1. Thus, this is the reason why in modulator development, with its systemic administration, sweat chloride has become really the go-to CFTR biomarker. The work by Edith Zemanek at the Sweat Chloride National Resources Center and others at that center has really advanced our understanding of sweat chloride through the design of a study called the CHECK-SC study, which I'll outline here, which is really looking at a longitudinal and observational measurements of sweat chloride on patients before and after the administration of modulators to understand the variability of response and the additive nature of sweat chloride reduction with various modulators. In the phase 1 study, those investigators were able to show a significant understanding of the variability of responsiveness in both patients on and off modulators, as shown by this work, and perhaps more importantly, have begun to understand the role of sweat chloride as a biomarker of additive effects of various modulators. For instance, in the left, switching from lumacaftor ivacaftor to tezacaftor ivacaftor for patients who are eligible for that drug, which demonstrated a incremental improvement in sweat chloride reduction. And more recently, the addition of triple or ETI therapy to patients that were previously on ivacaftor, demonstrating an additional reduction of sweat chloride, which is important biologically and is the kind of observations that are important for this type of large-scale sweat chloride study, and will continue to make this an important biomarker in the future. Importantly, because of this work, sweat chloride, if we'd ever develop systemically administered genetic-based therapies, may become back to the foray as a key biomarker for CFTR activity. Nasal potential difference has also been a key biomarker, especially in the early modulator development period throughout the first wave of CFTR trials in the early 2010s. At that time, the NPD assay was not standardized for this purpose. And so our group at the Center for CFTR Detection has been on a multi-year plan to improve the standardization of this technique, much like it's been done with sweat for decades before that. And by way of reminder, the NPD has been a key outcome measure for the early modulator developments, including ivacaftor, Adalir, and lumacaftor, with some of this data shown by work by Steve Rowe from the ivacaftor phase 2A trials going back some years ago. NPD standardizations have come across by several different levels, including standardization of electrodes, change to minimal perfusion systems, which minimize bubbling and other electrical interruptions, a standardized setup and experimental conditions, and standardized data acquisition, all in the last 10 years, with multiple publications coming out of our group, amongst others, in collaboration with many others across the US and Europe who have been involved in this space. These NPD methods, including and most notably electronic data acquisition and improved stability with a non-perfusion catheter system, have improved stability and reduced signal to noise and number of artifacts, which have overall improved the ability to detect. More recently, our group, in collaboration with many others who are experts in this field, have begun to identify scoring systems which standardize analysis of nasal potential difference. We noticed that several types of outcomes for NPD are viewed by experts as less interpretable and or have lower confidence in the measurements, as shown by the graphs in C and D compared to A and B. This allowed us to define a strictly analytical criteria to improve the assessment of reliability of data for NPD, which we published more recently. And using this in a multi-reader study, we're able to show significant inter-reader agreement of the qualitative score amongst several expert CF readers. When looking at the quantitative comparison of high versus low confidence tracings, for instance, we found that we were able to detect significantly higher discriminant ability for CF versus non-CF and the indeterminate range in high confidence tracings due to improved signal to noise ratio. More recently, NPD has been a primary endpoint in the first of many genetic-based therapies, including the antisense oligonucleotide elufericin, which was published a few years ago by Isabelle Sermet and others in our group. This data using NPD with nasal administration of elufericin in F508 del CFTR showed a rescue of both the sodium-dependent portion of the nasal potential difference via ENAC and chloride-dependent potential difference measurements that are CFTR-dependent in this data set. Now, as you can imagine, because of the interest in the development of lung-based and lung-delivered genetic-based therapies, lower airway potential difference that can lead to this level of discrimination is especially attractive. However, as I'll show in the next few slides, it's particularly difficult to do and it's not very well standardized, unlike NPD, and certainly not to the level of sweat chloride. This is a setup that shows an adaptation that our center has used for lower airway potential difference that relies on much of the equipment used for nasal potential difference. However, it is particularly challenging, as I'll go through the next few slides. This is the room setup, and it requires a good bit of cart placement and a lot of help to get this going. This is our group setting up for nasal lower airway potential difference at our center most recently. Bronchoscopic placement leads to a settling of the catheter in the lingular or segmental bronchus with touching the epithelium, but it's difficult to achieve that throughout about a 10 to 15 minute procedure. This is an example of ideal data from a previous study that we did looking at disease manifestations in COPD and acquired CFTR dysfunction some years ago using the current sequence and the current methodology. As you can see, there's quite a good discrimination between patients who have CFTR dysfunction versus those that have normal CFTR function. However, achieving this ideal data is not trivial. Therefore, with some standardizations, which I'll discuss a few more in a moment, we have designed a normative data study, which is currently enrolling, although has been slowed by the COVID-19 pandemic. We are selecting populations for normative data assessment, including 10 non-CF, 10 CF patients on highly effective modulators and 10 CF patients not on modulators. As I said, this study is currently enrolling in a single center site sponsored by our NRC grant at UAB. The early and normative data from our previous investigation indicates that we can show appropriate levels of total chloride conductance, or the zero chloride plus isoproterenol potential difference change in healthy non-CF subjects. We hope to show a good discrimination in this ongoing study to build on this previous data in our experience. We also hope to develop a next-gen LAPD probe and system that would simplify this and allow for multi-center investigation. As we stated earlier, there are numerous challenges with LAPD implementation for larger multi-center studies in CF, including the fact that it's an OR procedure due to deep cough and deep sedation required. The catheter is derivative of the NPD catheter, which is not commercially manufactured. The agar method for the non-perfusion probe is more problematic due to the long catheter length. There's bronchoscopic guidance required, which requires multiple staff, and the epithelial contact to be maintained with the bronchoscopic manipulation is particularly challenging in inexperienced operators. Therefore, in collaboration with our long-term collaborators, Gary Tierney and others at the Wellman Center for Photomedicine at MGH, we have been developing a new probe and simplified setup for multi-center studies, which includes derivation from an intestinal potential difference measurement of a catheter that has extremely small diameter and long-term stable electrodes. It also has conventional optical coherence tomography image guidance to ensure placement, and is compatible with a smaller endoscope, which could be used in unsedated subjects and allows for a plug-and-play type of consult, and is amenable to smaller animal investigation for preclinical studies of genetic-based therapies. This is an example of one of the schematics of the currently developing LAPD probe. These are the components. You can see it's quite easy. It's an out-of-the-box type of technique, and the probe here is very small on the order of less than 2 millimeters in width. As you can see from this video, we can use the optical coherence image in the upper left to ensure placement, and then get adequate contact with the epithelium. This is shown here in the intestine as an example. But when we achieve adequate contact, we're able to measure very appropriate and discriminative nasal potential or lower airway potential difference measures. So our previous work with NPD has formed a rubric for us to undergo a international collaboration to improve LAPD for performance of proof of concept studies in nucleic acid and other genetic-based therapies. And so we hope to develop methods and SOPs, because there is no current SOP at the CFF or CTN for LAPD performance. We'll collaborate with multiple leaders in the CTN Standardization Committee, including Isabel Sermay and myself and other colleagues at UAB, to develop a collaborative SOP and hopefully publish this and then implement it on ongoing studies or those which are upcoming, which we hope to be able to be a biomarker for the future. And finally, the Center for CFTR Detection at UAB has been devising LAPD's training scheme. Once we have completed portions of our normative data set and have developed the newer methodology and standardized it, we will begin training multiple sites through a multiple level didactic session and hands-on experiments with both animals and humans to acquaint these sites to allow for a multi-center investigation for a gene-based or other therapy in the future. Now, we also want to understand the development of biomarkers, especially in vitro biomarkers for advancing therapies, both modulators to newer CFTR indications, as well as optimizing therapies for other patients. As many will be familiar, the human bronchial epithelial data from the ETI therapies has been very potent along this development program for showing preclinical CFTR activation with new and advancing modulator therapies. A variety of tissue sources are under investigation, including multiple studies, including one which I had the pleasure of co-leading, including the RARE study, which is looking at blood-derived iPSC cells, nasal cells for planar culture, and rectal biopsy for development of organoids, all of which are going into cryopreservation for future study for this type of investigation at the CFF lab under the direction of Martin Mensa. As many will be familiar from the work the Netherlands by the Beekman Group and others, intestinal organoids can show a significant swelling in a force-induced swelling assay, amongst others, in the presence of CFTR modulators. And more recently, this group has been able to correlate improvements of key clinical outcomes, including FEV1 improvements, to the area under the curve of the organoid swelling in this type of assay. Work in our group by Jennifer Gimbalot, which mirrors work undergoing in many other labs, has shown that nasal-derived organoids can similarly show correlations to both improvement of sweat chloride as well as improvements of percent FEV1 in nasal-derived organoids in various swelling assays, which mirror the work done by the Beekman Group and others. And they are adapting novel method assessments to account for residual function CFTR to account for this full spectrum of CFTR activation. More recently, the work by the Beekman Group and others in collaboration in the Netherlands has shown that gene editing-based therapies, including CRISPR, can be demonstrated very well in preclinical testing for amenable types of mutations like the R758X CFTR, shown here as one example. So in this work, clonal populations of organoid-based, of intestinal-based organoid cultures are exposed to the gene editing system, which previously contained R758X. And then the prime clone is looked at organoid swelling after the gene editing system has been in place, and growth of these organoids back after these prime clones are developed. As you can see, the prime clones compared to the patient controls show significantly improved swelling, both quantitatively and then qualitatively here on the far right, with these confocal images compared to wild type and compared to patient control. They have similarly shown similar work for editing of other nonsense mutations in the F508del. This is just but one example of the many types of investigations in the preclinical setting using these types of biomarkers by other groups. In addition, organoid-based responsiveness is being tested in multiple studies, including the HIT-CF study, to optimize modulator development for various patients with rarer type mutations. And so I would encourage you to continue following this data, especially as we emerge from the COVID-19 pandemic. Short circuit current measurements and measurements of CFTR fluid flow, including mucus hydration, in human nasal epithelial ALI cultures may also correlate to improvements in lung function with modulators, as data shown by the Debley group and also group work from our group at UAB out of the GOAL and GOAL extension studies, which show that both short circuit current measurements may correlate on an individual level to patients' improvement in sweat chloride and improvements in short and long-term FEV1, as well as improvements of downstream effects of CFTR activation, including mucus hydration measured by the airway surface liquid depth, which correlates moderately with improvements of lung function in this same set of patients. If interested in exploring this and to further standardized methods for nasal epithelial cell testing, John Brewington leads the National Resources Center, which was recently founded at the Cincinnati Children's Hospital, and it has ongoing enrollment of patients with rare CFTR subjects, as well as significant standardization of these methodologies, which we have employed. And I would encourage you to look at his work for further developments. Finally, iPSC-derived airway epithelial cells may be a novel model system to promote clinical testing of Trikafta. This is an example from our work in collaboration with Eric Sorcher and Rachel Linneman at Emory University and Brian Davis at UT Houston of patient-derived iPSCs from subjects enrolling in a current clinical study who then have iPSC-derived airway epithelial derivation and then testing of triple combination or other modulator combos. We've shown in a previous study that partial restoration of CFTR activity in these iPSC-derived airway epithelial cells demonstrates this proof of concept and can correlate to the improvements in individual improvements in sweat chloride and lung function. More recently, work from subjects with the N1303K CFTR showed in the iPSC system to have activation of CFTR despite conflicting data in the literature. Therefore, we have designed a study to test both the improvement of sweat chloride as a primary endpoint in patients with N1303K treated with ETI therapy, and correlate this with changes in sweat chloride and the iPSC cell response to further develop this as a potential biomarker for future clinical development. With that data in mind, I'll conclude the following. In vitro and in vivo CFTR biomarkers have been a key to the development of CFTR therapies. Optimization of in vivo biomarkers is ongoing to determine the ideal options for proof of concept trials, especially for gene-based therapies. The various in vitro biomarkers remain key for preclinical investigation in human tissue and therapeutic. And the optimization of the properties of these biomarkers is ongoing as surrogate endpoints for therapies in rare CFTR mutations and may be useful for the future for gene-based therapies down the road. Are we in the live?
I don't know.
We are. I think we are, yes.
This is Tony, and I think the questions are in the chat room and designated to everybody to go through. And I don't know who wants to lead that discussion or not.
Thank you. I will do so. Apologies, everyone. It was not clear from our screen here that we were actually live. My name is Jennifer Groski. I'm from the University of North Carolina at Chapel Hill. I'd like to thank everyone for joining us today for the live Q&A session for Symposium 21. I'm moderating the session. My co-moderator today is Jane Davies from Imperial College London. Unfortunately, she's in high demand and is speaking in another session right now, so she's unable to join us for this live session, but I very much appreciate her help throughout this entire process. And I know I speak for her when I say thank you to our speakers for such stellar presentations. Joining me today are Dr. Nicole Meyer-Hamlet from the Seattle Children's Research Institute, Dr. Christopher Short from Imperial College London, and Dr. George Marty Solomon from the University of Alabama at Birmingham. When I was invited to co-chair this session, I couldn't be more excited about this topic that we were asked to address. For those of us who are engaged in clinical care and clinical research, this is definitely a hot topic as we seek to progress beyond FEV1. And so with that, I'd like to open up the session, and I'm actually going to start with a question for Nicole, and ask if you wouldn't mind clarifying for us the difference between your surrogate endpoints and your clinical endpoints, and what it sort of takes to move one of those surrogate endpoints into the more clinical realm, and specifically what it would take for the FDA to approve a surrogate endpoint as a lead outcome measure in a trial.
Sure. Well, clinical efficacy endpoints are directly related to how a patient feels, functions, and survives. Many people may not know this, but FEV for us is a surrogate endpoint. It's a surrogate endpoint for mortality and morbidity, but it's not actually an approved clinical efficacy measure. If you look on the FDA's website, it will list FEV as a surrogate endpoint. We didn't actually get there with FEV with a formal validation process. We had enough data epidemiologically to really kind of get it into that zone as a surrogate. exacerbations is a little bit more linked to some, how patient feels, functions, and survives. And that's also sometimes why we have these definitions for exacerbations when it comes to clinical trials, rather than just physician decision, because the FDA wants to see, some symptom profile associated with those exacerbations. Now for a surrogate endpoint, that's a really good question, because in a rare disease setting, it's often hard to get all of the evidence you need to formally validate, an endpoint as a surrogate. But what it typically needs is what's called a bridging study. And it's not just one bridging study, but multiple bridging studies to show that the effect, the change in the biomarker or the surrogate accurately predicts the change that you're going to see in the clinical efficacy measure. So to do that, you need to measure both the surrogate and the actual the clinical efficacy measure in the same study. And you need data just, not only from the active therapy group, but from placebo groups to compile that data. So it's a little bit of a high bar, and there's precedence within at least the FDA to approve therapies based on unvalidated surrogate endpoints. And we could, potentially go that route as well, but it would take further discussion.
Great, thank you. All right, if you guys wouldn't mind upvoting any questions that you really want to see answered so that we can make sure to address those during the time that we have left. The next question is actually going to be directed to me. And the question is, I wonder if you can imagine what technology improvements are needed to make MR ventilation scans more feasible from a patient perspective. So it can be a quick session encounter, perhaps by giving up structural imaging, or is the magnet a fixed barrier for simplifying the technology? And so I think that's a really great question. I think that this is definitely one of the issues that we deal with in trying to recruit and enroll patients into MRI studies. So the real challenge is that there needs to be more technological improvements in MR protocols. And this, when we talk about MR protocols, we're talking about a series of sequences that is really based on the number of radio frequency pulses and the gradients that allow us to actually generate an image. And so doing all those sequences in a protocol is really what takes the amount of time that it is required to do an MRI. And so it really depends on what you want to see. If there are, MR physicists who are actively working on generating more efficient sequences that can scan patients in a radial pattern or a spiral pattern, and thus not require such a long time within the scanner. And so I think that there are definitely improvements that are on the horizon in terms of being able to enroll patients a lot easier into these types of studies. Thank you for that question. All right. So next question to Nicole again. Nicole, are you aware of how companies have been incentivized to share data for drug development and other diseases?
That's a great question. Some of the large pharma companies have developed their own data sharing platform. We have a couple of things working for us. The MJE guidelines for manuscript publication. If anybody has tried to publish a clinical trial recently, there is a data sharing requirement now that you have to put in there, whether you're an investigator for an investigator-initiated study or a sponsor. So that is a little bit of a pathway that encourages data sharing and data transparency. The other thing going for us is just the increasing wealth of data platforms being built to de-risk, data sharing. And for us, I think the vision is that if we were to create a clinical trial repository that complements the CF registry, this would be incentive enough for sponsors to sort of this give to get sort of idea that if we can de-risk contributions of data, whether it just be placebo data to that repository, the wealth of data that could be accessible for clinical trial planning or for use as synthetic control data, and give access to this whole new suite of clinical trial designs that we have not had to use before, but may become necessary in the future, that could become incentive enough as we move forward.
Wonderful, thank you. Well, the next question is going to go to Chris. Chris, is there a role for LCI 5 versus LCI 2.5 in either CF research or care? How does this loss in sensitivity compare with the potential gain in efficiency, i.e. as in a shorter test?
Yeah, that's a real great question. And it's probably different, the response I give for each different age group. So I'll start off in the preschoolers. So there's a study done at the Brompton Hospital, which saw that if the preschooler could get down to LCI 5, then around 95% of them could get down to LCI 2.5 anyway. So if you're looking at feasibility, then it doesn't really add too much feasibility that way. And then in mild disease, where we look at preschool, school age, the loss in sensitivity is really probably too much of a loss, because the most sensitive portion of the multiple breath washout test is probably in between that LCI 5 and then the LCI 2.5. So that little bit of tail we have. So the loss that we have there is probably not worth it. If we're trying to save time, it would probably be better to see if you can do 2 MBW trials rather than the standard 3, as long as the quality is sufficient. And then there would be a question in severe disease whether LCI 5 would be able to track interventions. And certainly, like I said in my talk, the main problems in severe disease is the time it actually takes. So that may be one route where we go down there. But then again, in severe disease, how much information are we getting from a multiple breath washout test anyway? And should we be using alternative tests such as fMRI and cardiopulmonary exercise testing? It might give us more functional and better predictive data. And then there's also a great paper by Sanja Stevanovic from Sick Kids in the Journal of Cystic Fibrosis in 2015, which really lays out the differences between different endpoints. So LCI 5, and they even did like LCI 10 and LCI 3 in comparison to LCI 2.5. And each time it seemed LCI 2.5 was the most powerful. And then if you look at the work we've just done from our lab as well, just adding an extra large breath at the end seems to give us much more information. So I would certainly say the trade-off, particularly in mild and moderate disease, is probably not worth it. But in severe disease, it may need a further bit of research thrown at it to see what the answer is there. But thank you for the question.
Great, thank you. All right, Marty, sweat chloride may have already reached its limit of sensitivity to test for more efficacious, highly effective modulator therapy. May eventually the sweat secretion test substitute the QPIT in clinical trials?
Yeah, it's a great question. I have to say my work is not directly looked at that, but I know that in the NRC directed at sweat, directed by Edith Zemanek and others, and several recent publications have looked more at sweat secretion rates as a different form of that with various imaging modalities. A recent paper came out about a year ago or so looking at that and found that the detection limit for variability across even a more milder spectrum, for instance, of CF dysfunction, was able to be detected better than by regular sweat chloride. So effectively identifying sweat chlorides in that kind of moderate range, that would be more for diagnostic purposes. However, I think there's going to have to be a lot more work to answer that question, but there certainly seems to be a detection limit that's maybe a little bit further and better distinguishing than regular sweat chloride by the standard Gibson-Cook, as you're alluding to, that may be useful in that patient population. So we approach a set of patients who are reaching further and further into the normal function of sweat chloride. But to be honest, I've not done enough work myself to say where that is at as far as current, as far as an endpoint.
Gotcha. And there's another question here for you. Is there a capacity at the National Resource Lab to freeze down cells for future theratyping for patients who are currently on modulators but are not doing well clinically?
Yes, that's actually already happening through two sources. One is the H&E NRC is doing that for the patients they receive through their theratyping work. And the second is that we have been doing bioreposition of various patient types through the PROMISE and GOAL extension studies and have a large biorepository of nasal cells, frozen down with that correspond with clinical data, both those that are high responders and low responders, and are looking on analyzing that data as a secondary analysis right now, which we hope to be publishing from the GOAL study soon, then PROMISE is an extension of that and in coming years as that study concludes itself. So yes, the answer is already happening, just more work to be done with the data.
Okay. Nicole, can you speak to what we can learn by combining trials with non-CF bronchiectasis efforts? I'm also interested in what we can learn from working with CFTR carrier participants.
All right, two very different questions. So let me tackle the first one. A very good question. So I guess the idea here would be, could we consider basket trials with the non-CF bronchiectasis population, maybe for, say, an anti-inflammatory? I think this is a great question, as we're trying to figure out what, CF disease is now post-modulator, and if we're moving patients to look more like non-CF bronchiectasis. Non-CF bronchiectasis is a very heterogeneous disease. I think there's a couple things that we need to consider that factor into that clinical trial strategy. The first is that we need to really understand what is happening with exacerbation rates. Exacerbations is sort of the primary endpoint for later phase non-CF bronchiectasis trials. So that would be the expectation if we were to do those basket trials. We've seen that our rates have gone from, an annualized rate of 1 down to, 0.3 with, in the clinical trials. And maybe, as we saw, there is a poster that showed the 96-week ETI follow-up that's down to 0.21. Now, granted, that's during COVID. We need things to settle down a little bit, but we need to identify if there are a group of patients still having exacerbations and how we could maybe enrich those trials. And if we were able to do that, that's one thing. And to find a group of patients that had similar exacerbation rates to a non-CF bronchiectasis population so that they're a little bit more homogeneous. The second factor is, I think, finding a therapy that does have some proof of concept of effectiveness in non-CF bronchiectasis before we were to add in CF patients. I think that's been the difficulty. Before we add in another heterogeneous group in, if we could see some phase 2 data that shows some clinical efficacy, I think that would provide a stronger case for doing that type of basket trial.
May I comment regarding that, Nicole?
Yes,
I work in that space a fair amount with the non-CF bronchiectasis, and I think your point about the exacerbators is important. I think the targetable phenotype, rather than a particular subset of bronchiectasis patients along etiology, appears to be those that have more mucus production and neutrophil elastase elaboration resulting in exacerbations. Mm-hmm. I'm sure you're aware there's an ongoing oral phase 3 study looking at a neutrophil elastase inhibitor, an indirect inhibitor, and that had a reasonably good phase 2 data on the efficacy standpoint. Of course, this purpose was safety, of course, but the efficacy was reasonable, and led it to the pivotal study it's undergoing right now. And I think on the inflammatory mediator world, I think that is where that could overlap pretty well, because the driving force of exacerbations seems to be fairly uniform around various forms of bronchiectasis to be that neutrophil elastase and protease pathway. And so I think that could be, for the patients that continue to exacerbate, they probably will have higher neutrophil elastase levels like the frequent exacerbating bronchiectatic patients. So that's where that's looked in. And the sponsor of the oral inhibitor I was mentioning is going into CF with a phase 2A, I think, or maybe it's a 1B. I can't remember exactly where it's going relatively soon. So if that shows promise, I could foresee that being a concept where that could work, where there's a reasonably well-known shared pathophysiology in the illness.
Great,
Right.
thank you. Did you have more to say, Nicole, about the carriers?
Oh yes, well, I was going to throw that one over to Marty, but I mean, I think it's a really great question, and I think this is one where we do need to think creatively and really engage with our community. We are really committed to developing multiple therapeutic options, and it's curious as to whether or not we could engage with CF carriers to help figure out, relative, dose proportionality, of, say, if highly effective modulators or safe, some preliminary safety data. So to do some early work in that population, while we know we can't take our modulator population off of their modulators, and so I think it's a really creative, development approach, but one that we need to consider, the ethical implications and engagement of our community, as well as, the scientific rationale for doing that. I'd be curious, Marty, if you have thoughts on that.
Yeah, I sort of agree. I mean, the problem is the carriers are a heterogeneous population too, right? Some carriers have CFTR related disorder, and some carriers are more normal than I am with my recent walking pneumonia I'm getting over right now, so I think the understanding that population and really which of those have a true CF spectrum illness and which of them don't will be important. And, understanding how we can, the endpoints for that are also pretty messy. As I was alluding to earlier, I'm on a guideline committee trying to revise those to try to figure out which of that spectrum of patients really fits in the CF spectrum, which of them doesn't. And I think you could define it by one of any endpoint or one of any diagnostic endpoint like sweat. And if they're sort of intermediate, and they might look like a highly effective modulator patient, but they may have no illness or not a severe enough illness. So I think it's a hard question. It certainly has a lot of merit from a conceptual standpoint, but what we do, I think we do have to define the population better. And maybe it is time to start including those patients in sort of observation studies we're doing in CF to really define that population. And that may help with overlapping studies like the bronchiectasis question, where there's probably going to be a lot of carriers of those types of mutations, as that's known to play a factor in potentially an etiology and idiopathic type bronchiectasis.
So, I'll throw another monkey wrench into that ethical component that you guys are mentioning. So one of the earlier symposiums today was talking about the underestimation of the diagnostic and treatment rates of CF in other countries. The question that was occurring in my mind during that symposium was about research infrastructure in those countries, and if there is anything that we could and should be doing to help include those types of patients into clinical research. And it's obviously, fraught with problems, because it's not just about, bringing the research to them or them to the research, but also the treatment options afterwards. But there's a huge untapped population out there that is, hurting for help. Nicole, you unmuted. Did you want to say something?
Yeah, no, I think your point is extremely important, and one that I wasn't able to fit into my talk. But I think, any infrastructure we develop in these countries for doing research, it has to be matched with the commitment for access to these therapies. And that is, absolutely critical in our pathway forward. And so, I wasn't able to attend that earlier session. But I think, to the extent that we can partner with, the sponsors, that's what I'm really curiously interested in, as to how, they can formalize that commitment early on so we can make these investments.
Okay, I'm going to send another question to Chris. You mentioned that subjects with more severe lung disease, the LCI test may take several hours or may not be the best assessment for them. Would you say that adult studies should avoid LCI in study activities for those with severe lung disease? And is there a certain FEV1 at which you feel LCI is too cumbersome?
Yeah, so it's a real good question and something we've looked at quite a lot. Unfortunately, there's not a perfect correlation with FEV1. So it's not a clean cutoff, let's say at 60%, that's when it becomes too cumbersome. So some people, even with a FEV1 of around 50%, their actual multiple breath washout and LCI time doesn't take too long, mainly because they have certain parts of the lung which are not communicating, so it finishes quicker. But yeah, certainly I think a few recommendations which the courts, those central overreading centres have given out, I think they would probably say around 60% is a decent cutoff for not doing it in too severe disease, just because it can take too long. And certainly from an operator point of view, it can be rather hard to keep watching every single breath for minutes and minutes on end. But the one thing I'll add to that is that new software update that we've seen on multiple nitrogen washout is that the longer tests should now be a little bit quicker. So it's remains to be seen how much of an impact that has. But yeah, it is a challenge and probably not recommended in very severe disease.
Can I tack on a question that I had about that? I'm wondering if LCI is really the best or only marker that you can get from your NBW study, and whether I know that there's some reports about Z-scores and whatnot for the LCI, but particularly for those more severe populations, is there another aspect of it that we can look at and sort of get at that same information?
Yeah, so this comes back to what I was saying a little bit earlier on about how probably the most sensitive portion is that last portion. So unfortunately, there's no real way to bypass that early stage of the test to get down to the sensitive part. There is emerging techniques which may not need a strict, you have to go down to a certain percentage of the start percentage. Some really great work from Oxford University is looking at modeling the actual washout over a 5-minute course. So you wouldn't need to do 3 trials necessarily. You'd be able to just do a wash-in, either with a different trace of gas such as methane, and you could wash that out with either room air or 100% oxygen, and it would give you a load of different parameters. So that's something, a collaboration we're doing with Oxford at the minute, which maybe have promise for those more severe patients.
That's very interesting. All right, Nicole, what are the steps we need to take to redefine pulmonary exacerbations for those on HEMT in a way that will make the rate of pulmonary exacerbation an acceptable outcome measure to the FDA? Enjoy that.
Yes.
I'm glad you got that easy question, Nicole. Thank God that was directed to you.
That is from Jennifer Taylor-Cowser, in case
Okay, thank you.
Thanks for great edit. Nicole, good.
Okay, good. I'm sweating a little bit. So, I mean, I think the first thing we need to do is recognize the incredible amount of data that we're going to get this year as we emerge out of COVID and how incredibly important it's going to be. I mentioned earlier how we need to just get a handle of what's going on post-COVID and understand what our exacerbation rates really are right now, and understand, who, may be having more exacerbations, what they look like. In terms of developing, before we decide that we actually have to go down a path of developing a new endpoint, because that is going to be very challenging. To go down that path, you need an endpoint that meets a few criteria. We need, if we were to redefine exacerbations, it has to be meaningful. So we would likely have to link it to something that is grounded more in our, traditional clinical efficacy measures somehow. When we established that exacerbations was meaningful. It was built on years of epidemiologic data linking exacerbations to morbidity and mortality. So we don't quite have that data yet, especially if we define a looser, definition of exacerbations. The second criteria is it has to be sensitive to, measure treatment benefit versus a placebo group, And so I think, and especially probably for modest effect sizes, not something quite large, it's going to have to be, 30% reduction. So it has to be sensitive. And the third criteria is it has to happen frequently enough so that we can design trials that are feasible. So those are the three criteria that we kind of have to think through when we would go about designing a new endpoint. But I would say let's just pause on that for a little, and that's probably a symposium for next year. And let's really dig into the data that we're getting this year to understand, what exacerbations are and how frequently they are happening as our patients, thankfully, will, return to more normal life as we roll out boosters and so forth, and get them down to the kids.
One thing I'd love to see is what is the reality once we're able to get more in-clinic data from what you're talking about on asymptomatic lung function declines. Maybe that could be a reasonable assessment. I mean, because so many patients just don't cough now, and if they don't cough, they just don't bring it to our attention, and so I wonder if those kind of less symptomatic events may be something that could be used. It could still be an easy assessment tool. I mean, you can't get so complicated. It requires bunches of complicated lab studies and whatnot, or doing an LCI in clinic every time to figure that out. If it's too complicated, it's not useful either. But if it could be something as simple as modifying what an exacerbation looks like, or what the signal looks like, has to look like,
right,
so it would be detected. But it's still an easy way of detecting it.
And the role of home spirometry measure, home collection, in this. And, I will say the Stop Peds trial is ongoing. They will be getting, a wealth of data help contribute to this, question as well moving forward.
All right. The next question I'm going to send to myself. Does hyperpolarized gas MRI require a breath hold? Asking because wondering about its application in infants and young children. That is a great question. Currently, yes, a breath hold is required. So the most experienced centers are doing it down to about age 5. There are studies looking at neonates and talking about those more rapid sequence acquisitions that I was referring to earlier, where they are able to have the infants free breathe. And so these aren't necessarily CF infants, but rather BPD or other related neonatal disorders. And so, particularly in places like Cincinnati and Toronto, those are studies that are being designed and are ongoing. But yes, as of right now, I believe that the lowest age is around age 5. Now, the fluorinated gas MRI that I was referring to, well, we haven't done any pediatric studies in that yet. It is certainly on the agenda because since that is a free breathing protocol, or has the patient taking multiple breaths, we can design it as a free breathing protocol. And that was one of the things that was attractive to that particular type of gas inhalation rather than the hyperpolarized gas. So thank you for that question. Marty, Jennifer Taylor Hauser has another one for you. Just when you thought you were off the hook.
Yeah, right.
Is there a role for NPD or LAPD as an outcome measure outside of inhaled nucleic acid therapies? For example, if a systemic therapy doesn't move sweat, but moves NPD and LAPD, is it worth continuing to develop it? And I like that question.
I think that's a wonderful question. I would love to say we know the answer. That the short answer is we don't know. Part of it is that there's not a lot of standardization of this technique outside of a couple of labs with a lot of data to say that it probably won't be much more sensitive than nasal potential difference will be, which we know had a rather non-elusive history as an endpoint, for CFTR modulation in the early kind of world of CFTR modulators some decade ago. So I think the answer is I'd love to say it's the case. I'm hoping it is because I've got some research that's looking at that in the non-CF bronchiectasis world, but I don't know the answer to that yet. And that work is actually looking at patients like we discussed earlier, the kind of fringe CF type patients, whether or not it can be more sensitive going into an organ of interest rather than that. Whether or not it can be an endpoint, the answer is right now we just don't know. Mm-hmm. So I think it's mostly relegated, and the focus from the foundational level and whatnot is to develop it towards an inhalational therapies, which we think will be pretty common amongst the gene-based and editing therapies that are coming around.
Great. All right, Nicole, any thoughts about inclusion of modulator-treated CF patients in early phase dose-ranging transient genetic therapies such as mRNA, LNP, novel AAVs, et cetera?
Yeah, I mean, I think this goes into the same umbrella as, the carrier question. In some ways, minus the scientific part of it, because it's how do we preciously use our population on who don't have access to modulators. We want to move multiple therapies through the pipeline, and so we want to really make sure we can do that efficiently and safely. And once those therapies, are available to those patients, it's, at a dose that, may, be of benefit to them. And so I think it's a really excellent question, when is the appropriate time for some of these therapies also to, consider development in those on modulators. We saw from, initial survey results from CF Community Voice that there is engagement of the, patient population on modulators to participate in these studies, even if they may not necessarily benefit themselves. And I think that just shows incredible commitment from our community. And I will put a plug in for the CF Foundation and collaboration with Drs. Manu Jain and Chris Goss. I'm sure Manu will be talking about this tomorrow, have established a genetic therapies working group that is comprehensively evaluating all of these issues, and including, how we best partner with our community to think about this next phase of clinical development. And this is one of the questions on that list.
Nicole, I was struck by your comment about the poster 555 and the comments from the community about interest in developing these various different types of treatments. And one thing that I'd like everybody to maybe weigh in on is that we know that interest doesn't always translate to participation, right? And so what do we think are the key factors that we can do to make participation in these trials easier without compromising our design or our outcomes that we're looking for? And maybe I'll see if Chris has any, wants to start with that one.
I just merely turn up and do the tests, unfortunately. Yeah, I don't really have too much to add on that, so I'll pass over to the clinical colleagues if that's okay.
Nicole?
Well, I mean, I think that there's, obvious barriers that we need to address. I think, the number one is building trust with our community. And I think, there have been, just an incredible amount of, community engagement opportunities through that the CFF has really hosted over this last year. And that's probably the number one, factor that we need to work on, is building trust with our patients as we go forward with this work. And then we saw from some of the survey data that I presented, there are institutional, barriers, whether it's, having weekend visits or, so forth. I think the other thing to consider is just the challenges we will face with, some of these with trials for the population without access to modulators. We will need to recruit and do referrals. And so really centers working together to facilitate that process is going to be another factor.
Right.
There's also a bit of I think there's a couple things lacking in the next types of studies we're going to be doing. One is there's not a benchmark. The modulators took off because there was a great benchmark early with ivacaftor. We just don't have that yet. And so it would be great if there could be a safe, effective benchmark of like a genetic-based therapy, for instance, that we could hang our hats on to get the community motivated, I think. And right now, there's a huge intellectual, trust barrier, lots of barriers, as Nicole was alluding to, that are going to have to be overcome. I know there's a lot of efforts. I'm sitting on a call tomorrow, despite NACF happening, regarding that particular factor, because there's a lot of unanswered questions. And then of the other types of therapies, like inflammatory, anti-inflammatories, there's a lot of, well, do I need this therapy even now? Is it going to be beneficial to me or infectious? And so there's just a lot of new questions to answer with patients that are different than having just this is another modulator, which I think many would jump on the kind of quickly onto. So we need some benchmarking. We need, better education for sure.
Well said. I think that takes us to the conclusion of this symposium. We're out of time, unfortunately. Thank you, everyone, for your active participation. And thank you so much to all the speakers. I learned a lot from all of you today, and I appreciate it. Bye.
Thank you all.